Carl Dijkstraat's lab and Rob Malenka's lab, which are both psychiatry, bioengineering, and neuroscience labs at Stanford. And right now, I'm in the hospital most of the time, so I'm a psychiatry resident. I've kind of flipped back and forth from the hospital and the lab. So I came to tell you a bit about that. Tell you about this rat, which, because Priya hasn't been here yet, I imagine you guys don't know that much about. Or maybe you do. Have you heard of optogenetics? Have you had anybody come by and talk about it? All right, great. Sounds to be good. I also wanted to tell you kind of a bit about my path to getting to where I am now, because it's kind of funny and unusual. So that in mind, this is me. And I grew up in Alaska. So I grew up in Fairbanks, Alaska, which is the middle of nowhere. And newspaper day says it's going to be 50 to 60 below for the next 2 weeks. So that's more than 100 degrees colder than it is here right now. So it's extremely cold. This is actually not me in the snow there. This is in the snow in Boston. But very happy because it reminded me of the snow in Alaska. After that, I was in Boston. I was at Harvard for college, and I spent a year working at MGH, a new lab, which I'll tell you about in a minute. And then from there, I moved to California. I've been at Stanford since 2008. So I did an MD and PhD there, and now I'm a resident. But I think the important thing is that I'm very happy in all of these pictures. I'm very happy with my career trajectory that I've had. Just want to give you a little background story of how I got here. So we start with Alaska. Oh, don't worry about it, come on. So my parents, my dad's from Alaska, my mom's from Iowa, and they're both hippies. And dad lived all over the world. My mom lived on a farm with people named Wolf and Bear, and they raised rabbits. And I moved to Alaska, and they met working on the pipeline. So I grew up hanging out with bears. These are some black bears down where I used to go salmon fishing and halibut fishing. This is my family. These are fish we caught. This is not an optical illusion. These are very large fish. Big enough you have to shoot them when they're in the water before you bring them on the boat. And we used to hang out and work for the state government, going around, doing some recon. This is actually an adventure we went on because, you guys seen Into the Wild, or do you know this story? Into the Wild, so the bus. So it's real. So this bus is out in the middle of nowhere, and after the movie came out, all these people wanted to go find it, and kept getting stranded out in the middle of nowhere, and the government had to go rescue them. And so my buddy was a legislative aide, and they're like, hey, can you guys go figure out what's going on with this bus? And so we didn't find it because there's not much snow that year, as you can see. Usually there's many feet of snow, so this was as far as we could get. And these are my buddies. I went to high school with all a bunch of hippies. So those are my Alaska days. After that, I went to college, I went to Harvard, but in between, I took a stop before I went to college, which I would strongly recommend all of you consider. As it turns out, Harvard has this, I just took it off their website this morning, where the admissions committee encourages admitted students to defer enrollment for a year, to travel, pursue a special project or activity, work, or just do something that doesn't have to do with studying, essentially. So I took them up on it, but I realized I needed cash. So I had a bunch of different jobs. This is the pipeline. I worked on the pipeline in Caribou. Worked at a web site developer. This is back in the early 2000s, so HTML is all there was. And as a cook at this place called the Salmon Bank, which is the worst job I've ever had. It's terrible. But it made enough cash that I got a train ticket called a Eurail ticket, which you should all check out. This is unlimited train travel in Europe for 3 months. So I just cruised around. I worked at hospitals and stuff. And so had a nice adventure, and then had to come back. So came back. I did go to Harvard. There I met 2 guys who were instrumental in, or 3 people actually. I forgot I put you in here. They were instrumental in me kind of getting where I ended up. So this guy doesn't know me, and he wouldn't know me if you showed him my picture or anything. But I took a class from him my freshman year of college. So I got to college, I didn't know what I wanted to do. I'd done some computer work. I went to a high school. I was out in the boonies in Alaska. So we had these freshman seminars. And so I took one from this guy, George Whitesides, who's a famous surface chemist. So he does a lot of polymer work. And it was all about new technologies in biosciences. And so this was a kind of turning point in my life in a big way where I was introduced to a bunch of things I knew nothing about, but found them very exciting. So I was broke again at this point, and so needed some money, also wanted to learn a bit more about biology, and so applied for something called a federal work-study job, which is where the federal government will pay you to work in a lab when you're an undergraduate. So I got a job in this man, Doug Melton's lab, which back in the day, and currently to some extent, is a human embryonic stem cell research lab. So at the time I worked there, there was a federal ban on money, federal money, taxpayer money for human embryonic stem cell research. So he was inspired and driven by both of his children having type 1 diabetes. So this is the kind of diabetes you're born with. It's immune mediated rather than acquired. And so wanted to figure out how he could cure diabetes in his children. He wanted to use stem cells to do that. And then Chad was a postdoc in the lab. So after you've done college and gotten a PhD, then you become a postdoc before you're a professor. So he was on his way to becoming a professor, and he was my mentor. And so I guess the take-home point of this slide is that I think it's very important to try to find people that you respect and who are well-known and something you might be interested in that help to give you experience as you make your way through college or through high school or on your way to wherever you want to go. Which brings me to today. So this is the final picture. It's in California, which is why I'm standing in front of a bunch of succulents. So came here to Stanford to do a PhD originally, and then signed on to the MD PhD program. And again, was lucky enough to meet some people who were instrumental. So Carl Deisserock, who's well-known in neuroscience and psychiatry for generative technology called optogenetics, which I'll tell you about in a minute. Ofer, who's a good buddy of mine, who did a lot of the work I'm about to show you, and then Charu, who was kind of along the same path. And then just to give you a brief background on MD PhD programs at Stanford and other universities, you get training in both basic science, so it's kind of the right side, or your right side, my left side, and then you also get training in medicine. And so you bounce back and forth between these. So it lasts a long time, it's like 8 years. You do 2 years in med school, then you go do a PhD, then you go back to med school. But the whole idea behind this program is to try to give people basic training in science, but in order to answer questions that they bring from the clinic. And so the idea is to be a bridge between basic science in the lab and then also the hospital, the clinic, to generate new technologies with a specific focus on curing disease. And so the big question that I'm interested in, a little part of, is what does it mean to have a brain disease? You know, if you have pneumonia, it means you have an infection in your lungs, but we don't diagnose it based on that generally. We diagnose it based on like having a fever and a cough and feeling really terrible. But we don't treat feeling terrible, we treat the infection. So in psychiatric disease, the problem is that in schizophrenia, you're seeing things, hearing things, you don't interact with others well, you're very paranoid. Maybe you have some language or memory deficits. But we don't know what the actual problem is. We don't know what the equivalent of the infection in pneumonia is in schizophrenia or really any other psychiatric disease. And we focus on treating the symptoms, but that doesn't mean we're actually treating whatever the underlying cause is, the underlying problem. And so that ties in to the next part. Once we know what's causing it, then how do we fix it? And so the reason we haven't gotten to the point in psychiatry and neurology as we have in things like infectious disease, pulmonology when it comes to pneumonia or heart problems, is that we can't answer the two major questions in science. And those are necessity. So does A have to happen to cause B? And sufficiency. Is A happening enough to cause B? So in the case of pneumonia, just to bring it back to my example again, does an infection have to happen in order to cause the symptoms of pneumonia, a fever? And is an infection enough to cause these symptoms? And the answer to both of those is yes. But in the case of psychiatric diseases, neurological diseases, we don't have the ability to ask these questions, or we didn't until very recently. And the reason is that the brain is extremely complicated. So these are two very famous drawings from a Spanish gentleman named Ramon y Cajal. This is the hippocampus. Sounds like hippopotamus, but it's not. It's the memory center of the brain. So we know that it's necessary to generate the memories. And this is a beautiful Purkinje neuron. If we were to rotate this particular neuron 90 degrees like this, it would be completely flat. It's like a fan. But our cerebellum, the region of the brain that's responsible for coordinating our movements, it's packed. So as you can see, these are extremely complicated. The Purkinje neurons make thousands of connections to other neurons. The hippocampus interconnects with nearly the entire brain. So asking questions about how particular problems in either of these systems or in any system in the brain underlie a particular disease state, you can see would be very complicated, even more so in a human, let alone in a mouse. So as far as trying to understand how the brain works, Francis Crick, famous as part of Watson and Crick in the discovery of the structure of DNA, he later went on to be interested in neuroscience, said that the major challenge in neuroscience is that we don't have the way to manipulate individual components of the brain without affecting the rest of them. So, if we think of the brain like a computer circuit, it would be taking an individual transistor and being able to manipulate it without changing the activity of all the transistors around it. Where are we? So, to kind of give you a more concrete impression of why that's a problem, if we look at two neurons, so 1 and 2, and we stick an electrode in and zap it. The problem is that with electrical stimulation, while we can manipulate neurons precisely in time, we can flip a switch very quickly, like thousands of times per second, we can't choose which neurons we're manipulating. So if we have an electrode in the brain and we flip it on and off, we're going to modulate both of these neurons. We can't just pick out cell type 2, for instance. If we use drugs, so we have lots of drugs that manipulate the brain, right? They're more selective, so we can pick out cell type 1 and manipulate its function without necessarily impacting cell type 2. You can think the timeline here is very long. It takes time for the drug to be taken up, it takes time for it to act, and then for it to be metabolized. So both of these have shortcomings. It's a picture of the brain. So just to kind of reemphasize the point I made with the Ramon Hall slide, the green is one type of cell, the red is another, and these are 2 of hundreds that are in this particular space. But just to show you how interconnected they are, how they interact with each other, they also project widely beyond this space. So the lab I work in has generated a new technology that addresses this question. It's now used by thousands of labs around the world. We spun off a company, and it's actually being used in a human clinical trial right now. And it's based on something that can actually be found in the local area. So this is a Google Earth picture I stole of the salt flats near San Jose, which are these kind of funny colored ponds, where they have a lot of salt, like a ton of salt. So it would be the equivalent of like taking seawater, which already tastes salty, and making it 10 times more salty. And there's a bacteria there called Halobacterium salinarium, which has a particular protein called bacteriorhodopsin, which lives in my poor drawing of the membrane. So this would be outside the cell, this would be inside the cell. And it's very special because it combines sensing an external stimulus with some sort of ion movement. So in this case, it's able to take light from the sun and turn it into the movement of protons across the membrane. So it pumps them out of the bacteria. So that's important because neurons work based on gradients of charged ions, including hydrogen, including sodium, and others. When we looked further, we found that there were lots of different organisms. So here's an algae right here moving around. It's a little video I took. That do the same thing. So some of them live in dirt, some live in ponds, some live in, I think that's the Dead Sea. But they all do the same thing, which is to take light and convert the shape of a molecule. This is vitamin A, which is actually half of beta carotene. So they convert a molecule from one state to another. So you can see this little bit here has changed its orientation. And that's with the absorption of a photon. So bacteriorhodopsin I talked about a moment ago, it absorbs a photon, it pumps hydrogen out of the cell. One called halorhodopsin pumps chloride into the cell, and it's an actual pump. So for each photon it absorbs, it takes one of these ions and it moves it a little bit. And then the one I'm going to show you in a moment, channelrhodopsin, is an actual channel. It's closed, and then when it absorbs light, it opens. So this is important because in neurons, the action potential, which some of you may be familiar with, is actually underlined by the movement of ions. So the neuron hangs out here at rest. When it spikes or generates an action potential, it's a beautiful coordination of movements of ions across the membrane. So this initial depolarization or becoming more positive is the movement of sodium into the neuron. And then the repolarization, becoming more negative, is the movement of positively charged potassium out of the neuron. So the idea is that we can take these light-activated ion channels and pumps from bacteria that live in your neighborhood, jam them into neurons, and then control their activity with light. And so that's exactly what we're able to do. So you can see here, this is a neuron that's expressing channel rhodopsin. Every time there's a pulse of light, it causes it to generate an action potential. And so now we're able to take neurons that we select because these are genetically encoded. So a moment ago I showed you this picture with all the green neurons and the red ones. We can pick out just the red ones. We can express this channel in it, channel rhodopsin that's light activated using a virus. And then in a mouse, in a live mouse, we can control the activity of those neurons. So this is in motor cortex, which you'll see in a moment. Once it's activated, causes the mouse to run in circles because we're directly activating the motor cortex neurons. We're forcing them to fire, which then forces the mouse to perform a behavior because we're directly controlling those neurons, leaving all other neurons unmanipulated, which is what Francis Crick wanted. And to come back to my original point about the two questions in science, necessity and sufficiency, what this is showing is the sufficiency. So causing neurons, specific types of neurons, to have specific types of activity is enough to generate a behavior. But we also wanted to look at the flip side of that coin, this necessity portion. I'm going to skip this slide. But to do that, we needed a way to turn neurons off. So coming back to our action potential, where positive things make neurons more likely to spike, and making the neuron more negative makes it less likely to spike, we turn to Neutronomonas pharaenopsis, which is found in Egypt and the Dead Sea, etc., which is a light-activated ion pump that pumps chloride into the cell. So chloride is negatively charged. If we take that chloride and force it to go into the neuron, it'll make the neuron more negative. The idea being then that if we inject current in just a normal neuron with no manipulation, but then inject current and give our light, it turns the neuron off. So if I were to show this in an animal, this is a worm, and again has our ion pump in its motor neurons. It's moving around in water. When we shine light on it, it paralyzes the worm because it directly inhibits the motor neurons which connect to the muscles. So again, to come back to my necessity sufficiency point, this is showing that the activity would be necessary because we're getting rid of it. And these are very basic kind of examples using motor behavior because it's very obvious to see, but kind of extending this a little bit further into the original question I posited about what are the changes in the brain that underlie psychiatric disease. You can think that if we were to extend these technologies and say, what happens to normal mice if we replicate some of the findings from human patients with autism spectrum disorders? Which of those changes that we see in humans are enough to generate similar behavioral deficits? Or in Parkinson's disease, where we have a lot of hypotheses about what causes Parkinson's disease, what happens if we look at the brain of a mouse that we've caused to have Parkinson's disease and try to fix it? Which of those changes are sufficient to fix the behavioral deficits? So that's why these technologies, one, are very powerful for understanding the brain, but two, are actually even more powerful for being able to potentially fix problems once we figure out what they are. So as just a brief kind of way to finish up here, coming back to autism spectrum disorders, one of the projects I was involved with was trying to replicate changes that are seen very widely in human patients that have autism in mice that don't. And so it's an interesting thing to do because the core deficits in autism are things like social interaction and language, which are things we think of as being uniquely human, but most animals replicate to some extent similar behaviors. Mother mice call out to young mice, mice call to each other. They also interact socially in a very cute way. And so if we take a look at this, these are what mice do when they're just meeting for the first time. So you can see this guy's got a little white thing in his head because we have a fiber optic cable implanted in the brain. He's investigating a new mouse over here he hasn't met before. This is without any change. in the next video I'll show you in a minute, what we've done is we've increased just the amount of activity slightly in one part of the brain, in a very small part of the brain that's about 1 cubic millimeter, called the medial prefrontal cortex. So if I turn that movie on, pause this one, you can, mouse, spoiler, mouse spends the whole time interacting with the other mouse. So this is the exact same mouse, the same one, after we've just changed the amount of activity in one part of the brain slightly. So he's over here now. I think what you can appreciate is that there's a profound difference in the way that this same mouse interacts with two different novel mice with just his normal brain activity, or where we made a slight change in the amount of activity in a specific part of the brain, which in a number of ways exactly replicates what's seen in patients who have autism. And what we found later is that if we make a small change to bring that activity down, simultaneously it actually restores social interaction. So we can look at necessity and sufficiency of that part of the brain's normal functioning in social behavior. And so this, just to give you the long perspective on how that might come around to actually impact the clinic and the hospital. So we patented that, and then we've licensed it to a pharmaceutical company that's now using it to look at different medications that might be useful for patients who have this kind of social interaction deficit, so specifically autism spectrum disorder patients. So that's what I put together. It's kind of a whirlwind tour of my life. From growing up in Alaska in the snow, going to Boston, getting kind of interested in something. As a luck of the draw, took a seminar and then ended up here where I kind of bounced between developing new biotechnologies for psychiatry and neuroscience, and then also treat patients in the hospital, which I didn't talk that much about, but I'm happy to chat more.
What does your typical week look like between the lab and the...
It's crazy. So my last 6 weeks have really sucked because I've been really busy in the hospital. But it's month by month it changes. So I did 2 weeks where I was overnight, and I was covering 60 psychiatric patients at the VA, which is a veterans hospital, which has 3 different locked floors. And then the last month I was in the hospital caring for adult patients who are in the hospital for whatever, non-psychiatric problems, pneumonia among them. This week I'm on outpatient medicine, so at 1 o'clock I have to run over to the VA to do a liver clinic, and then tonight I'll be in the lab, be slicing up a bunch of mouse brains. So I really do bounce back and forth. But the eventual goal and the typical career trajectory is that you spend 4 days a week in the lab and 1 day a week in the hospital or the clinic. And that can mean 2 weeks in a block, it can mean half a day a week, kind of divided up. It depends what kind of deal you cut with your department. But we'll spend most of my time in the lab, running a lab, and then 1 day a week-ish caring for patients. All right. Hey.
Do people ever reverse that, so spend more time in the hospital and less time in the lab?
You could. I think it's trickier to do, simply because if you want to run your own lab, then it's a lot of kind of overhead administrative work. So you got to find money, which is kind of a crappy part of having to do it yourself, which takes a lot of time. So finding grants, you got to hire people, you have to check in with everyone. You have students you're responsible for, you have to make sure they're not failing, things like that. You can split it a little more evenly, or if you're primarily a clinician, so surgeons do this sometimes.
Yeah.
I work with a guy who's a neurosurgeon, and he has a project going, so he has some techs that work for him, and then he spends less than I want to spend in the lab. But it's very flexible.
Okay.
So you can spend less time. Yeah, you can kind of split it and make it work.
Okay.
Anytime you deviate from the path, it's a little bit more work to figure out how to make it work, make it functional. But it's totally doable.
Okay.
Yeah. All right. Any other questions about life, Alaska, neuroscience, wild animals? All right, very. Oh yeah.
Oh, so how did you, what got you into the environmental field in the first place in Alaska, like working with conservation projects?
Oh, I see. So I worked full disclosure, I worked for an oil company, but I kept them from getting oil everywhere. So two things, like one is I like working with my hands, so I like building things, which was a big draw of the kind of bioengineering is I can kind of, in my mind, make it seem like I'm working with my hands, even though it's mostly pipetting, which if any of you have done before is a lot of thumb work. So I enjoyed that. I like being outdoors. It's great money. So it was more of an opportunity that presented itself because I knew a lot of people who were doing it and could get a job there. But that was like 12 hours a day, 7 days a week, 8 weeks at a time. So you're kind of essentially just trading time for money, which worked out well because I knew I was going to go like cruise around Europe on a train for a few months. Yeah, so it wasn't as much environmental, it was more just building things.
Oh, that's cool.
Yeah, it was fun, yeah.
Why did you choose to go into psychiatry instead of neuroscience?
That's a great question. So Priya Rajagopal, I think is her last name, who will come talk to you, also did an MD PhD. She did at Columbia, but then chose to go directly into science. And so she's doing 100% science. You'll hear her perspective. And you can make that choice. So, once you finish med school, you decide what kind of doctor you want to be, or if you want to be a doctor, if you want to just go into the lab. So I thought about that, and I considered just going to do pure science. But I really like this part about having both skill sets where you can move back and forth and try to actually move things from the lab into the clinic. And so my long-term trajectory is I want to work on pain and like what it means to have chronic pain, which is has a huge psychological component. And so there's a big opportunity there to take findings from the lab and move them directly into your own clinic. So for me, there's a big one value add by being able to go and have patients that I treat routinely and work on things that are big components of having clinical trials. There are these review boards. And two is I really like patients. I like patient care. Meeting people is really fun. You can make a huge difference in people's lives in psychiatry, even given the kind of limited tool set that is available. And so I really just enjoy that. And so I wanted to make sure to keep that as part of my life. But it's a personal choice, as you had asked earlier, can you split things different ways? And you totally can. So you can do 0% clinic and 100% lab, which is what one of your future speakers will talk about. And a lot of people do 100% clinic and don't do any more research, and that's fine too. Yeah.
So when we're learning about the brain, we like learn the stuff that we do know. What's like something that's like puzzling about the brain that we don't know, like at all?
Oh man, there are so many things that are puzzling about the brain. So one of my favorite quotes of all time is from this dude named Linus Pauling, who is at Berkeley. And his quote was, "Imagine how many students we've failed for not knowing what we now know to be false." And I think that describes the brain very well right now. Because neuroscience is I'll make it brief, is in a huge state of flux. And what we thought we knew is core tenets and core principles are being challenged. So I think, let's see, if I was to pick out things in particular, I would say two of the big unknowns right now, one is like how much does the cerebellum contribute to things beyond movement? Because there are a lot of theories that have to do with the cerebellum contributing things like social interaction, behavior, autism. And another one, just for my personal interest, is like, what does it mean to be in pain? I think is interesting, because you can think about, have you guys seen The Matrix?
Yeah.
Brain essentially creates the matrix for us, and so we can manipulate it in ways to get rid of pain, or it can create pain where none exists. So there's something called phantom limb syndrome, where people lose an arm or a leg, but they have crazy pain in that limb even though it no longer exists. And so that's exclusively matrix-like phenomenon in your brain, where it's generating pain where there's no possibility of it existing. Yeah. All right, is that the end of our time?
Yeah.
All right,
Yeah.
thank you all very much.
Thank you.
Thank you.